You ever just stop and think like, wow, seeing is pretty wild. Like, it just happens. You open your eyes and boom, there's the world. But, I mean, under the hood, it's this crazy complicated process, right?
Oh, absolutely. It feels so instant, so effortless. But that seamless experience, yeah, it's the result of this cascade of, well, intricate events.
Totally. So today, we're diving deep into that world. We're going to unpack the science behind how we see, specifically focusing on Chapter 10, which really digs into vision and visual perception.
Right, and I think what's so cool is, you're interested in understanding the core of it all, how it actually works, and that's
Yeah.
exactly what we're going to do.
You got it. No getting lost in the weeds, just straight to the good stuff, the stuff that makes you go,
Yes.
aha. So that's how it works. Ready to get those aha moments.
Absolutely.
Perfect. So we're talking about everything from the very nature of light itself to how our eyes capture it, that wild journey that visual information makes to the brain, and then the big finale, how our brain turns all that raw data into, well, the colors and shapes and forms we see every single day.
Right, we're going to illuminate the key principles. And I think you'll find some of the things we uncover are truly surprising, even a little bit mind-bending.
It is mind-bending stuff. Okay, so let's start at the very beginning. Light.
Yeah, light. It's not a thing, like a rock or something. It's more like it's energy, dynamic energy that's always on the move, traveling in waves.
Like sound waves.
Exactly like sound waves. And just like with sound, where different frequencies give us different notes, different wavelengths of light give us different colors.
Oh, right. So like the light waves that look blue to us are a different length than the ones that look, say, red.
Exactly. To be precise, we're talking about nanometers. A nanometer is 1/1000th of a meter, so incredibly tiny. And for humans, the visible spectrum, that's all the colors we can see, it spans roughly from 380 to 800 nanometers.
I see. So like somewhere within that range, there's a specific wavelength that looks green to us. That's pretty wild when you think about it.
It is. And to give you a broader perspective, the visible light we see is actually just this tiny sliver of a much larger spectrum. Think radio waves, microwaves, X-rays.
Oh yeah, the electromagnetic spectrum. I remember that from physics class.
Right. Figure 10.1 probably shows it, with visible light tucked in there between infrared and ultraviolet. So our sense of sight is tuned into just the small window of the entire electromagnetic show.
Fascinating. Okay, so we've got this energy, this light oscillating in waves, but how does our body actually capture it? I mean, what's the mechanism that turns those waves into, well, something our brains can actually understand?
Well, that's where our eyes come in, specifically a part of the eye called the retina. It's like the movie screen at the back of your eye where the visual image gets projected.
I see. So the light enters our eye, passes through the lens, and then hits the retina. What happens then?
On the retina, we've got these specialized cells called photoreceptor cells. They're the ones that actually do the work of capturing light.
I remember reading about those. There are two main types, right? Rods and cones.
You got it. Rods and cones. Each type has a different job. Rods, they're the ones that help us see in low light. So they're super important for night vision.
Makes sense. So when it's dark and we can still make out shapes, that's our rods at work.
Exactly. They're very sensitive to even faint light, but they don't give us much detail or color information. It's more about just seeing that something's there.
Gotcha. So what about the cones?
Cones are the daytime specialists. They give us our sharp vision, the ability to see fine details, and of course, they're responsible for color vision.
Oh, right. Color vision. So those are the ones that allow us to see the world in all its vibrancy.
You got it. Now, here's the interesting part. These rods and cones, they're not just spread out evenly across the retina. They're actually concentrated in different areas.
Really? So there are like rod zones and cone zones?
Exactly. The cones, they're packed together in this tiny spot at the center of the retina called the fovea.
Okay. So when we focus on something, we're moving our eyes so that the image falls directly on the fovea, where all those cones are.
You got it. That's why your central vision is so sharp and clear. Meanwhile, the rods are more concentrated in the periphery of the retina. That's why you might notice movement out of the corner of your eye, even when you're not directly looking at it.
Oh, that makes sense. It's like a built-in motion detector. So cool. Okay, so we've got light hitting these rods and cones, but how do they actually turn that light into a signal that the brain can understand?
Well, inside those rods and cones, we've got these special molecules called photopigments.
I remember reading about those. They're like light-sensitive chemicals, right?
Right. In the rods, the photopigment is called rhodopsin. And in the cones, there are 3 different types of photopigments. each sensitive to a different range of wavelengths.
Oh, so that's how we see different colors. Each type of cone responds to a different part of the spectrum.
Exactly. Now, when light hits these photopigments, it triggers a whole cascade of events inside the cell. It's a process called phototransduction.
Phototransduction.
Yeah.
Sounds complicated.
It is complex, but the basic idea is this. You have a molecule called retinol that's bound to a protein called opsin. Light hits retinol, it changes shape, and that kicks off a chain reaction that ultimately changes the electrical potential of the cell.
Okay, so the light signal gets converted into an electrical signal, and that's what gets sent to the brain.
Right, but it's not a direct connection. Those signals from the rods and cones first get processed by other cells in the retina by polar cells and ganglion cells.
I see. So it's like a relay race. The signal gets passed from one cell to the next, and it gets refined along the way.
Exactly. And these ganglion cells, they have these fascinating receptive fields.
Receptive fields. What are those?
A receptive field is the area of the retina that a particular ganglion cell responds to. And many ganglion cells have the center surround organization.
Center surround. Like a donut.
Kind of. Imagine a circle within a circle. Some ganglion cells get excited when light hits the center of their receptive field, but inhibited when light hits the surrounding ring.
So they're sensitive to contrast, to edges and boundaries. That's pretty cool.
It is. It helps highlight the important parts of the visual scene. Now, the axons of all these ganglion cells, they bundle together to form the optic nerve.
The optic nerve, right. That's the cable that carries all the visual information from the eye to the brain.
You got it. And as that optic nerve travels towards the brain, it reaches a point called the optic chiasm.
The optic chiasm, what happens there?
That's where some of the fibers from each optic nerve cross over to the opposite side of the brain.
So it's like a partial swap. Why does that happen?
It ensures that each hemisphere of the brain receives information from both eyes. So the left side of your brain processes information from the right visual field, and the right side of your brain processes information from the left visual field.
Okay, that makes sense.
Yeah.
So after this optic chiasm, where does the signal go?
Straight to the visual cortex.
Not quite. It first makes a stop in the thalamus, at a structure called the lateral geniculate nucleus, or LGN for short.
The LGN, what happens there? It's like a relay station, or maybe even a filter. It receives input from the optic nerve and does some further processing before sending the information on to the visual cortex.
I see. So it's like another step in the relay race. The signal gets refined even further.
Exactly. And from the LGN, the signal finally reaches its destination, the primary visual cortex, also known as V1.
Okay, so V1 is where the real magic happens, right? Where we actually start to perceive what we're seeing.
You got it. And here's something cool. The visual cortex contains a retinotopic map.
A retinotopic map. What's that?
It means that the spatial arrangement of things in the visual world is preserved in the way that information is laid out in V1. So, like if you were looking at a face, the part of V1 representing the eyes would be next to the part representing the nose, and so on. It's like a little map of what you're seeing projected onto the back of your brain.
That's amazing. So the brain is literally creating a visual representation of the world.
Exactly. And there have been some really clever studies demonstrating this, using techniques like radioactive labeling to see which parts of the brain are most active when someone is looking at something specific.
That's so cool. Okay, so we followed the signal all the way to the visual cortex. Now let's talk about how we see color.
All right, so there are a couple of major theories about color vision. One is the trichromatic theory, also known as the Young-Helmholtz theory.
The trichromatic theory, right? Something about 3 different types of cones.
You got it. This theory says that we see different colors because we have 3 types of cones, each sensitive to a different range of wavelengths. So one type is most sensitive to long wavelengths, which we perceive as reddish hues. Another type is most sensitive to medium wavelengths, which we perceive as greenish hues. And the third type is most sensitive to short wavelengths, which we perceive as bluish hues.
Okay, so by comparing the activity of these 3 types of cones, our brain can figure out what color we're seeing.
Exactly. It's like mixing paint. By combining different amounts of red, green, and blue light, you can create the whole spectrum of colors.
That makes sense. But there's another theory too, right? Something about opponent colors.
Right, the opponent process theory. This one says that our color vision is based on pairs of opposing colors: red-green, blue-yellow, and black-white.
So like you can't see a color that's both red and green at the same time.
Exactly. And there are cells in the visual system that seem to support this idea. Some cells are excited by red and inhibited by green, while others are excited by green and inhibited by red.
Interesting. So which theory is correct?
Well, it turns out they're both kind of correct. The current understanding is that trichromatic processing happens at the level of the cones, and then that information gets fed into these opponent process mechanisms later on in the visual pathway.
I see. So it's a two-step process. First, the cones encode the basic information about color, and then that information gets further processed into these opponent channels.
Right. And those opponent channels are also important for enhancing color contrast. Remember those center surround receptive fields we talked about earlier? Well, some ganglion cells have color opponent receptive fields.
So, like, a cell might be excited by red light in the center of its receptive field and inhibited by green light in the surround.
Exactly. That helps make edges and boundaries between different colors stand out even more. Now, what about when something goes wrong with color vision? Have you ever heard of color blindness?
Oh, yeah, of course. But I don't really understand what causes it.
The most common cause is a lack of one or more of the photopigments in the cones. So, someone might be missing the red sensitive pigment, for example.
So they wouldn't be able to see red.
They would still see light in that part of the spectrum, but their brain wouldn't interpret it as red. They might see it as a different color, or they might have trouble distinguishing reds and greens from each other.
I see. So color blindness is basically a problem with how the brain interprets the signals from the cones.
Exactly, and it's much more common in men than in women because the genes for the red and green pigments are located on the X chromosome.
Oh, that's interesting. Okay, so we've got light and color down. Now, what about form? How do we see shapes and objects?
Form vision builds on a lot of the principles we've already discussed, like edge detection and contrast sensitivity. Remember those center surround receptive fields in the ganglion cells?
Yeah, they're sensitive to edges and boundaries.
Right. And as the signal moves from the retina to the visual cortex, it gets processed by increasingly complex cells. There's some really amazing work by Hubel and Weisel that showed how this works.
I think I remember reading about them. They discovered something called feature detectors, right?
Exactly. They found cells in the visual cortex that respond to specific features of the visual world, like lines and edges.
So like one cell might respond to a vertical line, while another cell responds to a horizontal line.
Right. And they found different types of feature detectors. Some respond to stationary lines, while others respond to moving lines. Some respond to lines of a specific length, while others respond to lines of any length.
Wow. So it's like our brain is breaking down the visual world into these basic building blocks, these features, and then putting them back together to create a representation of what we're seeing.
Exactly. And there's another theory called the spatial frequency theory that suggests the visual system also analyzes scenes in terms of their spatial frequencies.
Spatial frequencies, what are those?
Think of it like this. Imagine a picture of a zebra. It's got lots of high spatial frequencies because of the sharp transitions between the black and white stripes, but a picture of a clear blue sky would have mostly low spatial frequencies because the color is pretty much uniform.
I see. So our visual system is analyzing both the basic features of the world and the overall patterns of light and dark.
Exactly, and this processing happens in a very organized way. The visual system is highly modular, meaning that different parts of the brain are specialized for processing different types of information.
Oh yeah, that makes sense. We've already talked about how the left and right hemispheres of the brain process information from different parts of the visual field.
Right. And within the visual cortex, there are also different areas specialized for processing different types of information. There's a pathway called the ventral stream, which is often called the what pathway.
The what pathway? So that's how we recognize objects.
Exactly. This pathway goes from the visual cortex to the temporal lobe, and it's involved in identifying what we're seeing. Damage to this pathway can lead to a condition called agnosia, where a person can see an object but can't recognize what it is.
Wow, that's fascinating. So like they might see a fork but not know that it's a fork?
Right. And then there's another pathway called the dorsal stream, or the where pathway.
The where pathway. So that's how we figure out where things are in space.
Exactly. This pathway goes from the visual cortex to the parietal lobe, and it's involved in spatial awareness and guiding our movements.
I see. So like if I want to reach for a cup of coffee, my dorsal stream helps me figure out where the cup is and how to move my hand to grab it.
You got it. And within these pathways, there are even more specialized areas. For example, in the temporal lobe, there are areas that are specifically involved in recognizing faces.
Really? So there are cells in our brains that are dedicated to recognizing faces.
Exactly. And damage to these areas can lead to a condition called prosopagnosia or face blindness, where a person can't recognize faces, even the faces of their loved ones.
Wow, that must be incredibly difficult.
Yeah.
So our visual system is this incredibly complex network of specialized areas all working together to create our perception of the world.
Exactly. And it's all based on these three fundamental principles: inhibition, modularity, and hierarchical processing.
We've talked about modularity, how the visual system is broken down into these specialized modules, and hierarchical processing, how information flows from simpler areas to more complex areas. But what about inhibition? What's that all about?
Remember those center surround receptive fields?
Yeah, where a cell is excited by light in the center and inhibited by light in the surround.
Right. That's an example of inhibition. It's essentially the brain's way of sharpening the signal, of highlighting the important information and suppressing the less important information.
I see. So it's like turning up the contrast, making the edges and boundaries stand out even more.
Exactly. And inhibition is happening all throughout the visual system, not just in those ganglion cells. It's a fundamental mechanism that helps the brain make sense of the world.
Fascinating. Okay, so we've talked about light, color, and form. But our visual world isn't flat, is it? We see depth. How does that work?
Depth perception relies on a combination of cues, some binocular and some monocular.
Binocular cues, meaning we need two eyes to see them.
Right. One important binocular cue is retinal disparity.
Retinal disparity. What's that?
It's the slight difference in the images that each eye sees because they're in slightly different positions.
Oh, I see. So like, if I hold my finger up in front of my face and close one eye and then switch eyes, my finger seems to jump back and forth a little bit.
Exactly. That's retinal disparity. And the brain uses that difference to calculate how far away an object is.
That's pretty cool. What about monocular cues? What are some examples of those?
Monocular cues are things we can see with just one eye. Some examples are relative size, linear perspective, and interposition.
Okay, let's break those down. Relative size, that's just that closer objects appear larger.
Right. And linear perspective is the fact that parallel lines appear to converge in the distance.
Oh yeah, like when you're standing on a railroad track and the tracks seem to meet at the horizon.
Exactly. And interposition is when one object partially blocks another object. The object that's blocked appears to be farther away.
Gotcha. So our brains are using all these cues, both binocular and monocular, to create a sense of depth.
Exactly. And it's all happening unconsciously. We don't have to think about it.
That's amazing. And finally, there's this idea of active vision. What's that all about?
Active vision refers to the fact that we don't just passively receive visual information. We actively explore the world with our eyes.
So like our eyes are constantly moving, scanning the scene, focusing on different things.
Exactly. And these eye movements are controlled by a part of the brain called the superior colliculus.
We talked about the superior colliculus in the episode on movement, right? It's involved in orienting our attention.
Right. And in the context of vision, it helps us direct our gaze to the most important parts of the scene.
So like if something suddenly moves in our peripheral vision, the superior colliculus helps us quickly shift our eyes to focus on it.
Exactly. And these eye movements are crucial for building up a complete and detailed representation of the world.
Wow, we've covered a huge amount of ground today. From the nature of light to the intricate workings of the brain, it's incredible how all these pieces fit together to give us the experience of sight.
It truly is. And it's all happening so seamlessly, so effortlessly, that we often take it for granted.
Absolutely. But hopefully this deep dive has given you a newfound appreciation for the complexity and wonder of vision.
And perhaps it's left you with some lingering questions, like considering how much processing is going on behind the scenes, how much of what we see is actually a construction of our brains.
Right. Like, are we really seeing the world as it is, or are we just seeing our brain's interpretation of it?
It's a fascinating question to ponder, and it might make you look at the world around you in a whole new light.
I think it definitely will. Thanks for joining us on this deep dive. It's been a real eye-opener.
The pleasure was all mine.
Until next time, keep those aha moments coming.